So now let's move on to active transport mechanisms. So in the beginning we said that active transport mechanisms are those which require energy, and in these substances move against their concentration gradient. And these were the types of active transport mechanisms, that is primary active transport, secondary active transport, and there is another one for large molecules, that is exocytosis, endocytosis, and then again one is there which is penocytosis. We will consider all of them in active transport. Okay, before we move, let's consider some characteristics of active transport, basically of primary active transport and secondary active transport. So, as said that it transports substances against electrochemical gradient, and because it uses a carrier protein, it transports substances at faster rate, utilizes energy, that is ATP. It is mediated by carrier protein, so it will follow saturation kinetics, it will transport substances at faster rate, and it will follow competitive inhibition as well, and obviously it will be specific and inhibited by substances which block ATP generation or utilization, because it is using ATP, isn't it? So any substance which blocks ATP generation or utilization will block all active transport mechanisms. And hence it is of two types, primary active transport and secondary active transport. So let's consider an example of primary active transport, that is sodium potassium ATPase. Now remember, the proteins which are used for primary active transport, these are known as pumps. Now this sodium potassium ATPase which we are considering, it has binding sites for sodium as well as the potassium. Now you know that sodium is less inside the cell, so this is the intracellular fluid, right? And potassium is less outside the cell, so this is a extracellular fluid. Okay, so it is transporting sodium from inside the cell, that is less sodium concentration, to outside the cell where there is more sodium concentration, and similarly for potassium it is transporting from outside to inside. Inside has more potassium concentration. So both substances are moving against concentration gradient in this example. So primary active transport, even if it transports one substance or two substances, all the substances move against their concentration gradient. Fine. So in this example what happens is that there are three binding sites for sodium on the inside and two binding sites for potassium on the outside of the protein. When two potassium bind on the outside and three sodium ions bind on the inside of the protein, then this protein uses an ATPase which is also part of the protein. So this ATPase will break down ATP to ADP and phosphate, which will provide the energy, and then these substances will move. So this protein will flip actually, and the substances will move to the outside where they will be released. So that is the mechanism of action of primary active transport. There is other mechanism by which active transport works, that is secondary active transport. So let us see how this secondary active transport differs from primary active transport. Now secondary active transport is itself of two types. So first let's see co-transport or symport. So to understand this, let's consider one example. So here is the membrane. This side is showing extracellular fluid compartment and this side is showing the intracellular fluid. Now you know that sodium is always high outside the cell while inside it is low. Fine. And here we are considering one more substance, that is glucose molecule. Now glucose molecule here we are showing less outside the cell and high inside the cell. Okay. So we have to transport glucose from outside to inside the cell. So glucose has to move from its lower concentration to its higher concentration. Now we have shown another pump here, that is the sodium potassium ATPase pump, which we have already considered in primary active transport. So what this pump is doing is that it is continuously throwing 3 sodium ions outside the cell and bringing in 2 potassium ions inside the cell. So because of this pump, what happens that always sodium ions will remain high outside the cell. Now this carrier protein is SGLT, that is sodium glucose transporter. This has binding sites for sodium as well as for the glucose. So sodium will also bind and glucose will also bind. When both of them bind, it will transport these substances from outside the cell to inside the cell. So what is happening? Sodium is moving from its high concentration to low concentration, but glucose is moving from its low concentration to higher concentration. That means sodium is moving along its concentration gradient while glucose is moving against its concentration gradient, isn't it? So this carrier is, what it is doing, it is symporting or co-transporting sodium ion and glucose ions. And why it is known as symport and co-transport? Because both substances are moving in the same direction. Though their concentration gradient, if we consider the direction of the concentration gradient, it is opposite. Sodium is moving along concentration gradient, but glucose is moving against concentration gradient. But both are moving from outside to inside. So that is why the term co-transport, symport. Fine. So where is the energy utilized in this process? We said that active transport mechanism uses energy, but here we have not shown any use of energy. Yeah, actually secondary active transport utilizes the energy which is stored in the gradient of the sodium. So since sodium is moving from high concentration to low concentration, there is some potential energy stored in this, isn't it? So that energy it is using for transport of glucose, right? But where is the use of ATP? Well, you see, to maintain the gradient of sodium, this sodium potassium ATPase is continuously working. So basically the secondary active transport is indirectly using the energy. Actual energy is being utilized here for the creation of the gradient for sodium, right? So secondary active transport uses the energy indirectly, while actual energy is being used by primary active transport for maintaining the gradient of sodium, right? So this is secondary active transport. So example is sodium glucose transporter.